Imagine for a moment that extraterrestrial life has already arrived on our planet. They did not come in silver spacecraft descending from the sky, nor are they hiding in secret government facilities. Instead, they are flourishing in the depth of our own oceans. Picture a creature with 3 hearts pumping blue blood through a completely boneless body. A being that possesses a network of neurons distributed across 8 independent limbs, allowing its arms to taste, touch, and solve problems on their own. It can change its colour, shape, and physical texture in mere milliseconds, vanishing into the ocean floor like a phantom. This is not science fiction. This is the octopus. When evolutionary biologists map the tree of life, the octopus stands out as a glaring anomaly. It is a biological paradox, vastly separated from its closest relatives, like snails and clams, by millions of years of bizarre evolutionary leaps. It possesses a level of intelligence and physical capability that defies conventional logic, relying on genetic mechanisms that seem to break the fundamental rules of molecular biology. Its existence raises profound questions about the limits of life and the possibility of divergent intelligence. It is so remarkably different from mammalian life that studying it is the closest we can get to examining an alien organism without ever leaving our atmosphere. Today, we're going to explore why octopuses do not belong on Earth. We will dive deep into their extraordinary evolutionary history, their distributed nervous systems, and their mind-bending genetic code to understand how this creature became the ultimate outlier of nature. If you enjoy exploring the strange and fascinating realities of our universe, please like this video and subscribe to the channel. And before we dive into the deep, leave a comment letting me know where in the world you're watching from and what time it is for you right now. Let us begin. To truly understand why the octopus seems like an alien dropped into our world, we must first rewind the clock. We have to travel back in deep time, long before the first dinosaur took a breath, to an era over 500 million years ago known as the Cambrian period. This was a time of violent, rapid biological experimentation, a chapter in Earth's history where the blueprint for almost all modern animal life was frantically being sketched out in the primordial oceans. Out of this biological crucible emerged the phylum Mollusca. Today we associate mollusks with creatures like garden snails, oysters and clams. Slow, methodical animals whose primary survival strategy is to hide inside a hard, impenetrable rock of their own making. And for hundreds of millions of years the ancestors of the octopus did exactly the same thing. The ancient predecessors of the octopus were not soft, shape-shifting phantoms. They were heavy armoured dreadnoughts of the sea. Known as early cephalopods, creatures like the massive orthocones dominated the Paleozoic oceans. They constructed gigantic cone-shaped or coiled external shells made entirely of calcium carbonate, a chemical compound they meticulously extracted from the surrounding seawater. These shells were magnificent structures, operating not just as armour, but as complex buoyancy engines. By continuously regulating the balance of fluids and gases, such as nitrogen and oxygen, within the intricate internal chambers of their shells, these ancient cephalopods could float and manoeuvre. For millions of years, this design was the pinnacle of marine engineering. The heavy shell provided an absolute defence against the predators of the time, allowing these ancient cephalopods to become the apex predators of the Ordovician and Silurian periods. But evolution is never static. It is a relentless, unforgiving arms race. Fast forward to the Mesozoic marine revolution. The oceans were undergoing a terrifying transformation. A new generation of predators was entering the stage. Jawed fish with terrifying bite forces and hyper-agile marine reptiles, like the ichthyosaurs, began to prowl the waters. The rules of survival had suddenly and violently changed. Against these new, lightning-fast, bone-crushing predators, heavy armour was no longer an advantage. It was a death sentence. The massive calcium carbonate shells of the ancient cephalopods made them slow, cumbersome, and highly visible. They were outmaneuvered and easily crushed. A heavy shell is also exceptionally costly from a metabolic standpoint. It requires a constant, massive input of energy and raw materials to build and maintain. In the face of this overwhelming predatory pressure, the cephalopod lineage faced a brutal choice: adapt or face complete annihilation. It was during this intense period of evolutionary pressure that the ancestors of the modern octopus made a radical, seemingly suicidal leap. They began to abandon their armor. Over millions of generations, in a divergence that created the subclass Coleoidea, the external shell was gradually internalized. For the ancestors of the squid and the cuttlefish, the shell became a small internal support structure, the pen or the cuttlebone. But the ancestors of the octopus took this extreme evolutionary gamble to its absolute limit. They completely discarded the shell altogether. Today, the only remnants of their armoured past are two microscopic rods of cartilage buried deep inside their tissue, known as stylets. From a traditional biological perspective, giving up your primary physical defence mechanism when predators are getting faster and deadlier seems completely counterintuitive. It goes against the standard logic of survival. How does a soft, protein-rich animal survive in an ocean filled with razor-sharp teeth without any armour or bones? The answer lies in the profound biological trade-off that this shell loss unlocked. By ditching the heavy calcium carbonate fortress, the octopus freed itself from immense physical and metabolic constraints. Without a rigid shell, they were no longer bound by a fixed physical shape. They gained infinite flexibility. A creature that could once only float ponderously in the open water could now squeeze its entire body into the tiniest, most inaccessible rocky crevices on the ocean floor, slipping away from predators that were too large or too rigid to follow. But the most astonishing consequence of losing the shell was not physical, it was neurological. A hard external shell severely limits the internal spatial volume of an organism. It creates a strict biological ceiling on how large an animal's internal organs, specifically its brain and nervous system, can grow. By removing the shell, the octopus effectively removed the biological ceiling on its own intelligence. Without physical armour to rely on for protection, the octopus was forced to survive using its wits. It had to become infinitely aware of its surroundings, capable of outsmarting predators rather than just outlasting their bites. This intense evolutionary pressure ignited a cognitive explosion. The energy that was previously wasted on secreting calcium carbonate was aggressively redirected toward building massive, complex neural networks. The octopus traded a physical shield for a mental one. It developed high-definition, camera-like eyes to scan the environment, and a brain capable of rapid processing, learning, and memory. Consider for a moment the profound divergence between the octopus and its evolutionary cousins. The clam and the oyster chose the conservative path. They kept their shells, prioritizing passive defense. As a result, they remained mostly stationary, filter-feeding organisms with a nervous system so primitive they barely possess what we could call a brain. They are evolutionary survivors, but they are prisoners of their own armor. The octopus, by contrast, threw away the armor and walked into the dark, dangerous ocean completely naked. In doing so, it forced itself to become a genius of the animal kingdom. This dramatic evolutionary shift, however, creates a massive headache for paleontologists. To study the history of life on Earth is essentially to study hard biological materials. Paleontology relies on bones, teeth, claws, and shells, materials rich in calcium and phosphorus that can withstand the devastating forces of geological time and fossilize into rock. Because the modern octopus entirely lacks a skeleton, possessing only a hard chitinous beak used for crushing prey, it leaves virtually zero trace in the fossil record. When an octopus dies, its soft tissues decompose rapidly, dissolving back into the ocean ecosystem within days. It is a biological ghost. Therefore, tracing the exact timeline of their evolution requires incredible luck. paleontologists must rely on exceptionally rare geological formations known as lagerstätten, where perfectly anoxic conditions, environments completely devoid of oxygen, allowed soft tissues to be preserved as delicate carbon films in the rock before they could decay. In these incredibly rare deposits, scientists have found fossilized cephalopods hundreds of millions of years old, complete with the shadows of their 8 arms, their internal ink sacs, and even the suction cups on their tentacles. Some of these fossils are so perfectly preserved that scientists have actually extracted fossilized melanin from the ancient ink sacs, mixed it with ammonia and water, and used it to draw pictures of the very animal that died millions of years ago. These rare glimpses into deep time confirm a staggering truth. By the time the first dinosaurs were just beginning to walk the earth, the octopus had already mastered its alien form. It had already lost its shell, developed its massive brain, and armed itself with ink and camouflage. The great discard of the calcium carbonate shell was the definitive turning point in their history. It was the moment they stopped playing by the standard rules of terrestrial biology. By sacrificing their physical armor, they unlocked a pathway to a bizarre, hyper-flexible, and highly intelligent existence that looks, functions, and behaves unlike anything else in the natural world. They became a completely new kind of life form, setting the stage for the deeply bizarre anatomical anomalies we're about to explore. Having abandoned the heavy protective fortress of a calcium-based shell, the ancestors of the octopus were left with a profound biological problem. They were entirely soft. In the terrestrial world, a large animal without bones would simply collapse under the relentless pull of gravity, reduced to a helpless, immobile puddle of tissue. Even in the buoyant environment of the ocean, moving with speed, precision, and raw physical power requires leverage. To rip open the shell of a crab or overpower a struggling fish, a predator must be able to generate immense mechanical force. Invertebrates like us, this is achieved through a rigid skeleton made of calcium and phosphorus. Our bones act as solid levers, and our muscles pull against them to create movement. Insects and crustaceans achieve the same goal with a hard outer exoskeleton. The octopus, however, has neither. Aside from a small, parrot-like beak, its entire body contains 0 rigid structures. Yet, it possesses terrifying strength. It solved the mechanical problem of movement by engineering one of the most fascinating biomechanical systems in the natural world, the hydrostatic skeleton. To understand how an octopus moves, we have to look to the physics of fluids. The fundamental principle driving the octopus's anatomy is that water is essentially incompressible. You cannot squeeze a closed volume of water into a smaller space. The octopus weaponizes this law of physics by building its arms entirely out of densely packed, 3-dimensional arrays of muscle fibers that enclose a fixed volume of fluid and tissue. This structure is called a muscular hydrostat. While humans actually possess small muscular hydrostats, our tongues are built on the exact same principle, capable of complex movements without any bones. The octopus has scaled this concept to create an entire skeletal system. In an octopus arm, the muscle fibers are arranged in 3 distinct orientations. Longitudinal fibers running the length of the arm, transverse fibers radiating outward from the center, and helical fibers wrapping around the arm in a spiral pattern. Because the internal volume of the arm cannot be compressed, contracting one set of muscles forces an immediate and powerful expansion in another direction. If the octopus wants to extend its arm forward to strike at prey, it forcefully contracts the transverse muscles. This violently squeezes the internal fluid, making the arm thinner, which instantly forces the fluid outward, elongating the arm with the speed of a whip. If it needs to pull a heavy object toward its mouth, it contracts the longitudinal muscles, shortening the arm and generating massive pulling force. By utilizing the helical muscles, it can twist, bend, and articulate its arm at literally any point along its entire length. There are no joints to restrict its motion. An octopus arm possesses an infinite number of degrees of freedom. This architectural masterpiece of water and muscle grants the octopus an almost supernatural physical fluidity. Because it is completely unbound by the rigid architecture of bones or shells, an octopus does not have a fixed physical shape. It is, for all practical purposes, a liquid organism. This allows a creature weighing 50 pounds and stretching over 10 feet long to pour its entire body through an opening no larger than a small coin. When squeezing through a pipe or a crack in a glass tank, the octopus simply redistributes its internal fluid, flattening its organs and stretching its muscle hydrostats until it slides through the gap. The only absolute physical limitation on an octopus is its beak. Made of chitin, a tough biological polymer similar to human fingernails, the beak is the only part of the animal that cannot bend or compress. If a hole is large enough for the beak to pass through, the entire rest of the massive fluid body can follow. But manipulating a massive bone-free body through the heavy resistance of ocean water demands an extraordinary amount of metabolic energy. The muscles must fire constantly, and the brain must calculate millions of micro-adjustments every second. This high-performance biological machine requires a relentless, massive supply of oxygen to fuel its cells, and this is where we encounter the deep alien chemistry of the octopus circulatory system. If you were to draw blood from a human, or a dog, or even a fish, you would see the familiar crimson red. This colour comes from hemoglobin, a complex protein that uses atoms of iron to bind and transport molecules of oxygen. Iron is highly efficient in warm, oxygen-rich environments, making hemoglobin the standard respiratory engine for the vast majority of vertebrate life. But the octopus evolved in deep, dark, freezing abysses where oxygen is scarce, and the temperature hovers near absolute zero. In these extreme conditions, hemoglobin actually fails. The iron holds onto the oxygen molecules too tightly in the freezing cold, refusing to release them into the surrounding tissues where they are desperately needed. To survive this, the octopus evolved an entirely different chemical pathway for respiration. Instead of hemoglobin, their blood is packed with a respiratory protein called hemocyanin, and instead of using iron to capture oxygen, hemocyanin relies on atoms of copper. This copper-based biochemistry fundamentally alters the physical properties of the animal's blood. When hemocyanin is deoxygenated, having delivered its payload to the muscles and organs, it is completely colourless and transparent. However, the moment this clear fluid reaches the gills and binds with fresh oxygen from the seawater, a rapid chemical oxidation occurs. The copper reacts and the blood instantly turns a brilliant deep alien blue. While hemocyanin is brilliantly adapted to keep an animal alive in freezing low oxygen deep sea trenches, it comes with a severe biological penalty. Unlike hemoglobin, which is neatly packaged inside billions of tiny red blood cells that slide easily through our veins, hemocyanin floats freely in the blood plasma as massive, complex protein structures. This makes the blue blood of the octopus incredibly thick and heavy. It is a highly viscous fluid, flowing more like cold syrup than the thin, watery blood of a mammal. Pumping this thick, heavy blue blood through miles of microscopic capillaries requires an enormous amount of mechanical force. A standard single-pump heart would rapidly fail under the immense pressure required to keep this viscous fluid moving. To prevent its circulatory system from stalling, evolutionary engineering provided the octopus with 3 distinct, incredibly powerful hearts. 2 of these hearts are specialized pumps known as the branchial hearts. They are located on the outer edges of the animal's internal cavity, right at the base of its 2 gills. Their sole purpose is to gather the depleted, transparent blood returning from the extremities and forcefully drive it through the fine, delicate tissues of the gills. This high-pressure injection is necessary to force the thick blood to absorb oxygen from the surrounding water. Once the blood is charged with oxygen and turns dark blue, it flows inward to the center of the body, entering the 3rd and largest pump, the systemic heart. This central engine takes the freshly oxygenated blue blood and blasts it outward with incredible force, driving the heavy fluid back into the massive brain and the sprawling muscular hydrostats of the 8 arms. This 3-hearted, copper-fueled engine is a marvel of evolutionary adaptation, but it contains a critical, fascinating flaw. The systemic heart, the central pump responsible for keeping the body alive, is highly sensitive to extreme internal pressure. When an octopus wants to swim rapidly, using a method called jet propulsion, where it forcibly ejects a blast of water out of a muscular tube called a siphon, the violent contraction of its body cavity squeezes its internal organs with massive force. This muscular pressure is so intense that it literally crushes the systemic heart shut, forcing it to stop beating entirely. This means that whenever an octopus engages in rapid swimming to hunt or to flee from a predator, its main heart goes into complete cardiac arrest. It is forced to cut off the oxygen supply to its own brain and muscles. As a result, jet propulsion is catastrophically exhausting for the animal. It is sprinting while holding its breath on a cellular level. This physiological limitation explains a key aspect of octopus behavior. They absolutely hate swimming for long distances. Given the choice, they will always prefer to crawl along the ocean floor, gracefully dragging themselves over the rocks and sand using their muscular arms. By walking instead of swimming, they keep their internal pressure low, allowing all 3 hearts to pump in perfect, uninterrupted rhythm, sustaining their massive brains with a steady flow of copper-rich blue blood. The anatomy of the octopus is a masterclass in extreme biological workarounds, a creature built on principles of fluid mechanics and heavy metals, perfectly setting the stage for the most terrifyingly complex part of its biology, a nervous system that does not operate from a single mind, but from 9. In the architecture of terrestrial life, the nervous system is designed as a strict dictatorship. For mammals, birds, reptiles, and fish, evolution overwhelmingly favoured a top-down command structure. The brain sits securely in the armoured vault of the skull, acting as the undisputed central processing unit of the organism. It receives sensory input from the eyes and skin, processes that data, makes a conscious decision, and then sends a rapid electrical command down the spinal cord to the rest of the body. In this paradigm, the limbs are completely subservient. Your arms and legs are essentially organic mechanical tools, awaiting explicit orders from the central mind to execute any action. But when we plunge back into the ocean and examine the octopus, we find a neural architecture so deeply bizarre and radically decentralized that it forces us to reconsider the very definition of what a brain actually is. The octopus operates not as a biological dictatorship, but as a vast, interconnected, parallel processing internet of flesh. To grasp the scale of this alien intelligence, we must first look at the raw hardware. An average adult octopus possesses approximately 500 million neurons. This is a staggering monumental number for an invertebrate. It places their raw neurological processing power on par with that of a dog, a creature we widely recognize as highly intelligent, trainable, and emotionally complex. However, the way the octopus organizes and distributes these 500 million neurons is entirely alien to the vertebrate world. If you were to look inside the head of an octopus, you would indeed find a central brain, but its physical structure is deeply unsettling. It is shaped exactly like a donut. This brain is arranged in a thick neurological ring that tightly surrounds the animal's esophagus. This anatomical layout creates a terrifying physiological hazard. Because its brain wraps directly around its throat, an octopus must tear its food into microscopic pieces using its hard, chitinous beak before swallowing. If the octopus attempts to swallow a piece of crab shell or fish bone that is too large, the passing meal will physically bulge the esophagus outward, crushing the brain from the inside out and causing immediate neurological death. But the central donut brain is only a fraction of the story. Of the animal's 500 million neurons, only about 1/3 are actually located inside its head. The remaining 2/3, roughly 350 million neurons, are completely banished from the central executive brain. Instead, they are pushed outward, weaving a massive, dense neurological web down the entire length of its 8 appendages, This is not just a thick bundle of nerve endings meant to transmit pain or temperature, like the nerves ending in human fingertips. Each of the 8 arms contains massive, highly organized clusters of neurons that act as independent processing centers. In functional terms, the octopus does not have one brain. It essentially has 9. There is the central brain in the head, acting as an executive overseer, and 8 smaller, highly capable mini-brains running the entire length of each limb. Why would evolution build such a wildly fragmented mind? The answer lies in the sheer biomechanical nightmare of controlling a completely fluid body. As we explored earlier, an octopus arm has no bones and no joints. It is a complex muscular hydrostat capable of infinite degrees of movement. If a human arm, with its simple shoulder, elbow and wrist joints, requires massive central processing power to execute a simple task like catching a ball, imagine the computational power required to control an arm that can bend, twist, elongate, and contract at literally any millimeter along its entire length. Now, multiply that by 8. Add to this the fact that each arm is lined with hundreds of individual suction cups, each of which is highly prehensile and can move independently like a tiny muscular finger. If the central brain of the octopus had to consciously calculate every single microcontraction of every muscle fiber and monitor the pressure of every single suction cup across all 8 arms simultaneously, the biological processor would instantly crash under the sheer volume of data. The animal would be paralyzed by the infinite complexity of its own body. To avoid this catastrophic neurological bottleneck, the octopus evolved the ultimate system of delegated computing. The central brain does not micromanage the arms. It simply issues a high-level command, a broad, general intention. For example, if the central brain's visual cortex registers a crab hiding deep under a jagged rock, it does not calculate complex mathematical coordinates, detailing exactly how the arm should articulate to reach the target. The central brain simply sends a broadcast signal to a specific arm that roughly translates to, "Go catch that crab." Once that signal is sent, the central mind can completely disengage and look elsewhere. The arm itself takes over. The millions of neurons embedded directly within the limb calculate the geometry of the rock, analyze the resistance of the water currents, and coordinate the complex firing of the muscular hydrostats to snake around the obstacle, grasp the struggling prey, and bring it back to the mouth. The arm operates with total local autonomy, adjusting its grip and changing its physical shape in real time, without ever needing to consult the central brain in the head. To facilitate this, at the base of every single suction cup lies a massive cluster of nerve cells called a ganglion. Each sucker is essentially equipped with its own localized microprocessor, capable of registering tactile information and making immediate micro-decisions. The central brain is only kept informed of the general outcome of the action. This decentralized architecture creates an organism that experiences reality in a way we cannot possibly comprehend. For humans, cognition is physically locked inside our skulls. We are our brains, and our bodies are just vehicles we pilot. But for the octopus, cognition is physically smeared across its entire body. It is a creature whose physical limbs are not just tools for interacting with the world, but active participants in perceiving and thinking about it. This extreme biological distribution of intelligence blurs the line between the body and the mind, raising profound questions about what it actually means to be a conscious entity when your thoughts are literally occurring in 8 separate appendages at once. The implication of having 350 million neurons distributed across 8 limbs goes far beyond simple motor control. It fundamentally changes how the octopus perceives reality. For a human being, the senses are strictly segregated. You use your eyes to see, your nose to smell, your skin to feel, and your tongue to taste. But the octopus breaks down these sensory walls, because the massive neural clusters in its arms operate with intense local autonomy. The limbs do not just blindly grope in the dark, they actively smell, taste, and make complex decisions entirely on their own. To understand this, we must zoom in on the anatomy of a single suction cup. A large octopus can have over 2,000 suckers distributed across its 8 arms. These are not simple biological vacuum plungers designed merely for sticking to glass or rocks. They're incredibly sophisticated sensory organs. Each individual sucker is packed with tens of thousands of specialized chemoreceptors and mechanoreceptors. Mechanoreceptors allow the sucker to feel the physical texture and weight of an object, while the chemoreceptors are specifically designed to detect chemical signatures in the water. In mammalian terms, this means that every single suction cup on an octopus acts as an independent nose and tongue. When an octopus reaches its arm into a dark, narrow crevice on the ocean floor, it is not just feeling for the shape of a hidden crab. It is literally tasting the water and the rocks. If you could imagine having a tongue on the tip of every single one of your fingers, capable of tasting the salt on a pretzel or the sugar on a piece of fruit just by lightly touching it, you would begin to understand the sensory world of the octopus. As the arm slithers over the reef, the localized mini-brain inside the limb constantly processes a flood of chemical and tactile data, distinguishing between the taste of a poisonous sea sponge, an inanimate piece of coral, and the rich protein signature of a sleeping fish. And remarkably, it does all of this without needing to send that massive amount of sensory data back up to the central brain for analysis. The ultimate and perhaps most unsettling proof of this localized intelligence comes from controlled scientific observations of severed octopus arms. Because octopus limbs are frequently bitten off by predators like moray eels or sharks, the arms are evolutionarily designed to remain highly functional even when completely disconnected from the central nervous system. When a biologist surgically removes an arm from a heavily anesthetized octopus in a laboratory setting, the amputated limb does not simply lie dead and motionless on the table. It continues to live, react, and process information for hours. If a scientist gently pinches the skin of a completely severed octopus arm, the limb will immediately recoil, pulling away from the painful stimulus just as a living animal would. More astonishingly, if you place a piece of food, like a small chunk of fish, near the severed arm, the dormant suckers will suddenly spring into action. They will chemically taste the food, recognize it as prey, and firmly grasp it. Then, acting on pure localized programming, the suckers will begin to pass the piece of fish from one sucker to the next, moving the food steadily up the length of the severed limb toward the base, aiming precisely for where the central mouth used to be. The arm does not know the body is gone. The localized neural loop contains all the necessary coding to identify prey, capture it, and attempt to feed an organism that is no longer there. This terrifying level of autonomy introduces a massive logistical nightmare for the living animal. If you have 8 highly intelligent autonomous arms, all independently tasting the water, grabbing objects and making their own decisions, how do you prevent absolute biological chaos? What stops the arms from accidentally attacking each other or tying themselves into an inescapable knot while wrestling a chaotic, thrashing prey? The answer lies in a brilliantly elegant chemical fail-safe built directly into the animal's skin. The octopus utilizes a highly specialized mechanism of chemical self-recognition. The skin covering the octopus is embedded with a unique chemical signature, a specific biological protein marker that acts as a permanent identification tag. When an autonomous arm reaches out and a suction cup brushes against another arm, the chemoreceptors instantly taste this chemical marker. The moment the sucker tastes its own skin, a powerful localized inhibitory reflex is triggered within the arm's nervous system. This reflex completely shuts down the sucker's ability to grasp. It forcibly prevents the arm from latching onto itself. The localized mind of the arm effectively registers the chemical signal as a strict command. This is self, do not grab. This fail-safe operates entirely outside of the central brain's control. Scientists have proven this by observing that a severed arm will completely ignore another severed arm from the same octopus. However, if you strip the skin off an amputated arm and present it to another arm, the suckers will instantly grab the flesh and try to eat it. because the chemical warning signal has been removed. This decentralized, chemically regulated neural network transforms the octopus into something resembling a localized hive mind. It is a biological collective, an alliance of 9 separate processing centers that cooperate to form a single entity. The central brain acts as the executive board, analyzing visual data from the eyes and determining the overarching goals, such as deciding to hunt, mate, or hide. But the execution of those goals is completely surrendered to the 8 brilliant tasting, smelling, and thinking limbs. The octopus is not piloted by a single consciousness trapped inside a skull. It's an organism that literally thinks with its entire body, experiencing the physical world with a level of distributed awareness that is completely alien to the vertebrate paradigm. If a distributed nervous system that allows an animal to think with its arms is not bizarre enough, we must now turn our attention to the surface of the octopus. We must examine its skin. In the realm of terrestrial biology, skin is primarily a static barrier. It regulates temperature, keeps internal organs in, and keeps pathogens out. But for the octopus, skin is not just a protective wrapping. It is a highly advanced, neurologically driven, 3-dimensional optical display screen. It is an organ of deception so technologically perfect that modern military engineers are actively trying, and failing, to replicate its underlying physics. The octopus does not just hide from its predators, it literally bends the physics of light to erase itself from the visual spectrum. To understand how an octopus can vanish against a rock in less than 200 milliseconds, we have to look at the microscopic architecture of its skin. The ultimate weapon in the octopus's optical arsenal is a specialized layer of cells known as chromatophores. Imagine millions of microscopic, highly elastic balloons buried just beneath the surface of the skin. Each of these balloons is filled with a dense pool of biological pigment. In octopuses, these pigments come in 3 primary colors: black, brown, and a yellowish red. In a reptile like a chameleon, color change is driven by a slow, hormone-based system that shifts pigment around within the cells over a period of seconds or even minutes. The octopus does not have time for hormones. Instead, its camouflage system is wired directly into its high-speed electrical nervous system. Every single one of the millions of chromatophore balloons is attached to a radial array of microscopic muscles, and each of those muscles is connected directly to a nerve fiber leading back to the brain. When the octopus needs to change color, the brain fires a massive volley of electrical signals. The microscopic radial muscles forcefully contract, violently pulling the edges of the pigment balloon outward. The balloon stretches out, instantly transforming from a tiny invisible dot into a broad, flat disc of solid color. By rapidly firing different combinations of these black, brown, and red discs across its massive surface area, the octopus can generate shifting, complex patterns, creating stripes, spots, or solid, dark bands in the blink of an eye. When the danger passes, the muscles relax, the elastic balloons snap back into tiny dots, and the color instantly vanishes. But black, brown, and red are not enough to achieve perfect invisibility. The ocean floor is a riot of vibrant colors: bright pink corals, emerald green algae, and the shimmering blue of the water itself. To mimic these colors, the octopus utilizes a deeper layer of specialized cells called iridophores. Unlike chromatophores, which use actual physical pigments to absorb light, iridophores do not contain any color of their own. Instead, they rely on the physics of structural coloration. Inside an iridophore cell are complex stacks of ultra-thin protein plates. These plates are arranged at incredibly precise microscopic distances from one another, acting as biological prisms. When white sunlight penetrates the ocean and hits the octopus's skin, it passes through the top layer of chromatophores and strikes the iridophores. The protein plates refract the incoming light, bending the photons and physically splitting the light into its component wavelengths. By neurologically altering the angle and spacing of these microscopic protein plates, The octopus can actively tune the cells to reflect specific wavelengths of light back at the observer. This is how the octopus generates brilliant iridescent greens, metallic blues, and shimmering silvers, colours that are impossible to create with its standard pigment balloons. Finally, below the iridophores lies a third base layer of optical cells known as leucophores. These are flattened structural cells completely coated in crystalline purines. Leucophores act as perfect, broadband light scatterers. They are the ultimate biological white background. Whatever ambient light exists in the environment, whether it is the bright, unfiltered white light near the surface, or the dim, blue-shifted light found in the deep water, the leucophores automatically scatter it back outward with perfect fidelity. They ensure that the octopus always perfectly matches the ambient brightness and tonal baseline of its immediate surroundings. By layering these three cellular technologies, the muscular pigment balloons of the chromatophores, the light-bending protein prisms of the iridophores, and the ambient light-scattering crystals of the leucophores, the octopus operates a biological television screen. It dynamically mixes pigments and refracted wavelengths to match any background with terrifying precision. Yet true invisibility requires more than just a two-dimensional colour match. The ocean floor is highly textured. A perfectly coloured but perfectly smooth octopus sitting on a jagged reef of coral would still cast a shadow and look incredibly obvious. To solve this, the octopus employs yet another biomechanical trick. It manipulates the physical topography of its skin in real time using structures called papillae. Much like the muscular hydrostats that make up their arms, the papillae are concentrated bundles of erector muscles embedded throughout the skin. By tightly squeezing the water and tissue within these bundles, the octopus can force its skin to bulge outward in highly specific shapes. It can raise tall, jagged spikes to mimic a piece of rough kelp, create rounded bumps to look like coarse sand, or pull the skin completely taut to resemble a smooth, polished stone. In a fraction of a second, the animal changes not just its colour and brightness, but its physical, three-dimensional geometry. This brings us to one of the greatest, most baffling paradoxes in all of modern biology. An animal capable of instantly analysing a complex visual environment and flawlessly replicating the exact colours and textures of a coral reef must possess the most advanced, high-definition, colour-sensitive eyes on the planet. Right? Wrong. Extensive physiological studies of the octopus eye have revealed a staggering truth. While their eyes are incredibly complex, featuring a cornea, an iris, and a lens similar to a human eye, their retinas contain only one single type of photoreceptor. In vertebrate terms, they have no cones. From a biological and neurological standpoint, the octopus is completely and totally colourblind. They see the entire universe in shades of black, white, and grey. How is this possible? How can a creature that exists strictly in a grayscale reality perfectly mimic the vibrant reds, greens, and blues of its environment? How do you paint a masterpiece in full colour when you're physically incapable of seeing the paint? For decades, this paradox drove marine biologists to the brink of frustration. It defied all logic. The answer, discovered only recently, once again forces us to throw out everything we know about how biology is supposed to work. The secret does not lie in the eyes of the octopus. It lies in the skin itself. Scientists began to notice that even when an octopus was completely blinded, or when severed pieces of its skin were placed in a petri dish, the skin would still change colour in response to shifting light. Upon analysing the molecular structure of the skin tissue, researchers found massive concentrations of a specific light-sensitive protein called opsin. Opsins are the exact same photoreceptive proteins found in the retinas of the eyes. Their entire biological purpose is to capture incoming photons of light and convert them into electrical signals. But in the octopus, these opsins are not confined to the eyes in the head. They are distributed by the millions across the entire surface area of the animal's body. The octopus does not need to see colour with its brain. because its skin is essentially acting as a massive distributed retina. The skin itself can directly detect the wavelength, intensity, and angle of incoming light. When blue light hits a specific patch of skin on the octopus's left arm, the local opsins register the wavelength. This triggers a localized subconscious neural loop that automatically commands the chromatophores neridophores in that exact microscopic area to adjust their pigments and structural plates to match. The animal is not consciously looking at a yellow sponge, thinking about the colour yellow, and commanding its skin to turn yellow. The skin is physically reacting to the yellow light bouncing off the sponge in real time, operating completely independently from the visual cortex in the central brain. The octopus is literally seeing the environment with its entire body. This revelation shatters our understanding of sensory boundaries. For the octopus, the line between vision and touch is completely blurred. Its skin is simultaneously a protective layer, a tactile sensor, a chemical tasting organ, and a highly advanced optical receptor array. It is a creature that has decentralized not only its intelligence, but its very perception of reality. It feels the light and tastes the shadows, rendering it an absolute phantom of the deep and a masterclass in alien optical physics. We have explored the alien hardware of the octopus, its distributed neural network, its three-hearted circulatory system, and its shape-shifting, light-bending skin. Now, we must examine the software running on this bizarre biological machinery. When scientists study animal intelligence, they look for specific behavioral markers that indicate advanced cognitive processing. For decades, complex problem-solving and the deliberate use of tools were considered the exclusive domain of higher vertebrates. Primates like chimpanzees, highly social birds like crows, and marine mammals like dolphins. Invertebrates, programmed with simple biological algorithms, were not supposed to be capable of such feats. But the octopus once again completely shatters the established hierarchy of the animal kingdom. Its behavior in the wild and in the laboratory reveals an intellect so profound and so highly adaptable that it forces us to redefine the evolutionary boundaries of intelligence itself. To understand the depth of octopus cognition, we must travel to the sandy, barren ocean floors off the coast of Indonesia and observe a species known as the veined octopus, frequently referred to as the coconut octopus. In this specific marine environment, the seafloor is highly exposed. There are very few coral reefs or rocky crevices to hide in, leaving any soft-bodied creature highly vulnerable to predators. To survive in this desolate landscape, the coconut octopus has developed a behavior that sent shockwaves through the scientific community. It has learned to scavenge for discarded half-shells of coconuts or large clamshells and use them as mobile armor. When a veined octopus finds a suitable coconut shell, it does not simply hide under it like a hermit crab claiming a discarded snail shell. The octopus actively manipulates the object as a complex tool. First, it uses its powerful arms and localized intelligence to blast away mud and debris, thoroughly excavating and cleaning the hollow interior of the shell. If it finds two matching halves, it will stack one inside the other, exactly like a stack of nesting bowls. Then, it does something truly extraordinary. The octopus flips the stacked shells underneath its central body, wraps several of its arms tightly around the payload to hold it in place, and violently stiffens its remaining arms to lift itself off the seabed. Operating like a bizarre multi-legged mechanical crane, the octopus begins to walk clumsily across the ocean floor on the tips of its stiffened tentacles, carrying its heavy, awkward prize. This behavior, known as stilt walking, is incredibly taxing. It is slow, it consumes a massive amount of metabolic energy, and most importantly, it leaves the octopus temporarily vulnerable and exposed. So why do it? Because the octopus possesses a cognitive ability previously thought to exist only in the most advanced mammals: foresight. The animal is carrying the coconut shells not because it needs shelter right now, but because it anticipates needing shelter in the future. When a predator, such as a shark or a large predatory fish, eventually appears on the horizon, The coconut octopus stops walking. In a fraction of a second, it drops its payload, slips its soft liquid body between the two halves of the coconut, and violently pulls them shut from the inside using the powerful suction cups on its arms. It transforms instantly from a soft, exposed piece of protein into a perfectly spherical, impenetrable wooden fortress. The predator investigates, finds only an inedible coconut resting on the sand, and swims away. By sacrificing short-term safety and energy for a long-term strategic advantage, the octopus demonstrates temporal awareness. It can plan for events that have not yet happened, executing a deliberate multi-step survival strategy that requires an incredibly sophisticated internal map of cause and effect. This capacity for high-level problem-solving is further amplified when researchers bring the octopus into the controlled environment of a laboratory. When presented with artificial challenges, the octopus displays a terrifying capacity to understand mechanical systems. One of the most famous demonstrations of this is the jar-opening experiment. If a scientist places a live crab inside a heavy glass jar with a screw-top plastic lid and drops it into an octopus tank, the octopus does not merely attack the glass with blind predatory frustration. It evaluates the puzzle. Through its advanced visual cortex, the octopus identifies the prey. Using its highly sensitive suckers, it maps the smooth, impenetrable surface of the glass, quickly realizing that brute force will not work. It then investigates the seam between the glass and the lid. The suckers, acting as independent sensory arrays, feel the microscopic ridges of the screw thread. The octopus, wrapping its arms around the lid, coordinates hundreds of localized suction cups to grip the plastic perfectly, creating a massive amount of friction. Then, using the muscular hydrostats in its arms, it applies deliberate, calculated rotational torque. It twists the lid, unscrews it completely, discards the plastic, and calmly reaches inside to claim its reward. They can even reverse engineer this puzzle. If an octopus is placed inside the jar and the lid is screwed shut from the outside, the animal will map the internal geometry of the container, locate the structural weak point of the lid, grip the inside of the glass with half its body, and use the other half to push and rotate the lid outward, freeing itself. Their capacity for spatial mapping and memory retention is equally staggering. In complex maze experiments, octopuses are forced to navigate multi-level transparent plastic labyrinths filled with dead ends and false pathways to find a food reward. Not only do they rapidly learn to solve these physical puzzles through trial and error, but they also demonstrate long-term memory retention. If an octopus solves a complex maze and is then removed from that testing environment, it can be dropped back into the exact same maze weeks or even months later, and it will navigate the labyrinth perfectly on the very first try, without making a single wrong turn. In vertebrate brains, this kind of spatial learning and long-term memory consolidation is handled by a specialized neural structure called the hippocampus. The octopus, lacking any shared evolutionary history with vertebrates, has no hippocampus. Instead, it has evolved a completely different neurological structure called the vertical lobe, a massive, densely packed cluster of neurons dedicated entirely to learning and memory. Through the sheer force of evolutionary pressure, the octopus brain arrived at the exact same cognitive destination as the mammalian brain, but it took a completely different biological highway to get there. This is a phenomenon known as convergent evolution, and it proves that high-level intelligence, mechanical understanding, and future planning are not exclusive to animals with backbones. The octopus is a self-aware, strategic, and highly analytical thinker, capable of turning the objects in its environment into sophisticated tools to conquer the challenges of a hostile world. The intelligence of the octopus is not strictly limited to cold, calculated survival tactics or mechanical puzzle solving. When you place a highly advanced, massively parallel processing brain inside a confined space, you do not just get a biological machine operating on basic instinct. You get a personality. You get a creature that observes its environment, analyzes the other living beings within it, and acts with deliberate, often shocking individuality. To truly grasp the alien nature of their minds, we must look at how they interact with us, their mammalian captors, and the extraordinary lengths they will go to in order to entertain themselves or escape our containment. For a long time, evolutionary biologists assumed that to an invertebrate all humans looked exactly the same. We were perceived merely as massive, blurry, terrestrial shadows moving outside the glass. But extensive research in aquariums and marine laboratories has revealed a terrifyingly sophisticated cognitive ability. Octopuses can recognize individual human faces. They do not just see us, they study us, and they form distinct, long-term opinions about specific people. In controlled laboratory environments, researchers have conducted targeted behavioral experiments to test this. They designate one scientist as the primary caregiver, responsible for feeding the octopus high-quality food like shrimp or crab. Another scientist is designated as a mild annoyance, tasked with gently prodding the animal with a bristle brush or shining a moderately bright light into its enclosure. Both scientists wear the exact same uniform, stand at the exact same height, and do not speak, eliminating any auditory or broad visual cues. The octopus learns to distinguish between the two individuals with frightening speed. When the caregiver walks into the room, the octopus will immediately rise to the surface of the tank. Its chromatophores will shift into smooth, relaxed patterns of light brown or pale white, and it will extend its arms outward, actively seeking tactile interaction and food. However, the moment the annoying scientist enters the room, the animal's reaction is violently different. The octopus will instantly retreat to the darkest corner of its enclosure. Its skin will flash a deep, aggressive red, a colour universally associated with anger or warning in cephalopods. If the scientist steps too close, the octopus will aim its siphon, the muscular funnel normally used for jet propulsion and steering, and fire a high-pressure blast of cold salt water directly at the scientist's face. This behaviour is cognitively profound. It requires extreme visual acuity to map the microscopic geometric differences between two human faces across a pane of glass and through a distorting layer of water. It also requires the massive vertical lobe of the octopus's brain to store that complex visual data, attach an emotional response to it, and recall it instantly upon the person's return. This level of facial recognition and individual grudge-holding is a trait previously thought to be strictly reserved for highly social, pack-hunting mammals like wolves, dolphins, or primates. Yet the octopus, a solitary creature with zero evolutionary need for complex social hierarchies, possesses it entirely. When an intelligence of this magnitude is confined to a sterile laboratory tank, a new behavioural phenomenon emerges: boredom. Boredom is a luxury of the intelligent. A simple organism merely exists, reacting only to hunger or immediate threats. But a brain capable of spatial mapping, memory retention, and problem-solving requires constant neurological stimulation. When an octopus is understimulated, it does not simply sleep. It becomes maliciously mischievous. Aquarium staffs around the world possess endless frustrating accounts of cephalopod vandalism. If a bright halogen light bulb above a tank is irritating the animal's highly sensitive opsin-rich skin, the octopus will not just hide under a rock. It will calculate the trajectory, position its siphon just below the surface of the water, and shoot a precise high-velocity stream of water directly at the glowing hot bulb, shattering the glass and short-circuiting the entire electrical grid of the laboratory. They will systematically dismantle the intricate plastic valves of their water filtration systems, not because the flow of water bothers them, but simply to see how the pieces fit together, flooding the floors of the facility in the process. They have been observed taking floating plastic pill bottles and repeatedly bouncing them against the water jets in their tanks, catching them as they get pushed back, engaging in a solitary game of biological ping-pong just to pass the time. But their absolute highest display of cognitive superiority lies in their legendary prison breaks. Modern aquariums must treat large octopuses not as fish, but as maximum security inmates. Because of their hydrostatic skeleton, possessing absolutely no rigid bones other than their chitinous beak, they can exploit the tiniest flaws in human engineering. These escapes are never accidents. They are heavily premeditated, multi-step operations. Octopuses are known to observe the patrol routines of the night guards. They wait for the lights to go out and for the building to go perfectly quiet. Then they use the massive pulling force of their longitudinal muscles to pry open heavy tank lids, some of which are weighed down with heavy rocks or concrete blocks. Once the lid is open, the animal pulls its liquid body out of the water and onto the dry, hostile floor. The most famous of these incidents occurred at the National Aquarium of New Zealand, involving a male octopus named Inky. In the dead of night, Inky managed to force open a tiny gap at the top of his enclosure. He squeezed his entire body through the crack, slid down the glass, and dropped onto the floor. But he did not just wander aimlessly. He navigated across the large room, actively searching the perimeter until he located a small 6-inch wide drainage grate. He manipulated the grate, squeezed his massive basketball-sized body into the narrow plumbing, and crawled through 50 meters of dark, winding pipes that emptied directly into the Pacific Ocean. He was never seen again. In other facilities, staff have been baffled by the mysterious disappearance of rare, expensive fish from neighbouring exhibits, only to review the nighttime security footage and discover the culprit. An octopus had silently pried open its tank, slithered across the floor, climbed up into the neighbouring exhibit, hunted the fish, eaten them, and then quietly returned to its own tank, closing the lid behind it before morning. These are not the automated, hardwired reflexes of a lower life form. This is an organism that observes the architecture of its captivity, maps its surrounding environment, calculates risk, and executes elaborate, multi-stage heists. It is an alien intellect that fundamentally refuses to be contained by human boundaries, proving undeniably that profound awareness, curiosity, and mischief can evolve on a biological path entirely separate from our own. To truly comprehend the alien nature of the octopus, we must move beyond its physical anatomy, its distributed nervous system, and its bizarre behavioral intelligence. We must zoom in to the microscopic level and dive into the very instruction manual of its existence. We must look at its deoxyribonucleic acid, the fundamental genetic code that dictates exactly how this organism is built and operated. In 2015, an international team of marine biologists and geneticists achieved a monumental biological breakthrough. They successfully sequenced the entire genome of the California two-spot octopus. What they found hidden inside those microscopic strands of genetic material completely shattered the established rules of evolutionary biology. The genetic blueprint of the octopus is so profoundly bizarre, so radically complex, and so heavily scrambled that it looks almost nothing like the genetic code of any other animal currently living on this planet. First, we must evaluate the sheer, staggering size of the biological data. In the animal kingdom, genomic size generally correlates with physical and evolutionary complexity. The human genome, long considered the gold standard of biological sophistication, contains approximately 3 billion chemical base pairs. An average invertebrate like a garden snail or a fruit fly has a genome that is only a tiny fraction of that size, reflecting their simpler biological architecture. But when scientists finally measured the entire genome of the octopus, they found that it contained an astonishing 2.7 billion base pairs. It is virtually the exact same size as the human genome. For a completely soft-bodied marine invertebrate to possess a genetic instruction manual as massive and intricate as a mammalian apex predator is a glaring biological anomaly. It completely defies the standard expectations of invertebrate genetics. But the biological mystery deepens significantly when we look at how many individual functional genes are actually packed into that massive ocean of code. A gene is a specific sequence of deoxyribonucleic acid that contains the exact instructions for building a specific protein. Humans, with our massively complex brains, highly advanced adaptive immune systems, and intricate skeletal structures, possess roughly 20,000 to 25,000 protein-coding genes. The octopus, astonishingly, possesses over 33,000 protein-coding genes. It has significantly more individual genetic instructions than a human being. How does a creature that lives for only a year or two, possesses no rigid bones, and spends its entire life crawling on the ocean floor, require a genetic code that is mathematically more complex than ours? The answer lies not just in the raw number of genes, but in the chaotic, highly unusual way the octopus has chosen to arrange them. In standard evolutionary biology, species that share a common ancient ancestor usually share a similar gene order on their chromosomes, a foundational concept known as genomic synteny. If you compare the genome of a human to a mouse, or a bird to a reptile, you will consistently find long, unbroken stretches of identical genetic sequencing. Even across vast periods of evolutionary time, nature tends to keep successful, highly functional blocks of genes grouped closely together. But when scientists meticulously examined the octopus genome, they found absolutely zero genomic synteny with any other known organism on Earth, not even with its closest shelled relatives like oysters, clams, or snails. The genetic code of the octopus looks as though someone took a standard invertebrate genome, threw it into a high-speed industrial blender, and violently scrambled the resulting pieces across the chromosomes. This extreme genetic scrambling is actively driven by a massive, uncontrolled concentration of specialized genetic elements known as transposons, frequently referred to in biology as jumping genes. Transposons are essentially rogue pieces of deoxyribonucleic acid that possess the unique, somewhat dangerous ability to detach themselves from one location in the genome, copy themselves, and blindly insert themselves into completely different, random locations across the chromosomes. In the vast majority of complex animals, jumping genes are strictly suppressed and chemically silenced by the body because they cause dangerous mutations, cancer, and severe genetic instability. But the octopus has completely embraced this genetic chaos. Nearly half of the entire octopus genome is composed of these highly active transposons. By allowing these jumping genes to run wild, the octopus has essentially turbocharged its own evolutionary process, constantly shuffling its genetic deck of cards to create radical, unpredictable biological innovations. One of the most profound innovations born directly from this chaotic genetic blender is the massive, unprecedented expansion of a specific gene family known as protocaderins. To build a highly complex nervous system, an animal desperately needs a way for millions of individual neurons to accurately find each other and connect. Protocaderins are the critical biological glue that makes this possible. They are specialized proteins that sit on the outside of nerve cells and dictate exactly how those cells wire together to form a functioning brain. In the standard invertebrate world, neural networks are simple and centralized, so the genetic code only requires a few of these glues. A standard nematode worm or a fruit fly, for example, possesses only a single dozen protocaderin genes. But because the octopus abandoned its external shell and chose to build a massive, decentralized network of 500 million neurons distributed across 9 separate brains, it required a radically upgraded wiring system. Through hyper-accelerated genetic duplication, driven by its jumping genes, the octopus expanded its genome to include an astonishing 168 distinct protocaderin genes. This is a level of neural wiring complexity that is completely unheard of in any animal outside of the vertebrate lineage. Alongside the massive expansion of protocaderins, the octopus genome also features a terrifyingly large expansion of zinc finger transcription factors. These are highly specialized master control genes that dictate exactly when, where, and how other subsequent genes are turned on or off during the development of an embryo. While most invertebrate animals have a very modest collection of these control switches, The octopus genome contains hundreds of unique zinc finger genes, specifically dedicated to managing the highly complex localized construction of its shape-shifting camouflage skin, its powerful muscular hydrostats, and its highly autonomous thinking arms. The genome of the octopus is not merely a simple variation of a standard biological template. It is a wildly overgrown, highly unstable, and beautifully chaotic library of biological information. It is heavily armed with jumping genes that constantly rewrite the evolutionary rules of its own existence, and it possesses an arsenal of neural building blocks that perfectly rival the most advanced mammals on Earth. But as incredibly complex as their standard deoxyribonucleic acid is, it represents only the beginning of the genetic paradox. Because the true, undeniable alien nature of the octopus does not lie in how it statically stores its massive genetic code, but in the highly controversial, universally rule-breaking way that it actively edits that code in real time. The chaotic, scrambled nature of the octopus genome is astonishing, but it is ultimately a static map. To uncover the true, terrifying brilliance of their biology, we must look at how they actually use that map in real time. We are going to examine a biological process so radical that it essentially breaks the foundational law of life on Earth. In the scientific community, this law is known as the central dogma of molecular biology. It is the absolute rulebook that dictates how biological information flows within a living cell, and it applies to virtually every single organism on the planet, from the smallest bacteria to the largest blue whale. The central dogma is built on a strict sequential assembly line. It states that the permanent genetic code is stored within the nucleus of the cell as deoxyribonucleic acid. When a cell needs to build a physical structure, like a muscle fiber or a neural receptor, it cannot use the deoxyribonucleic acid directly. Instead, it makes a temporary disposable copy of that specific genetic instruction. This copy is called messenger ribonucleic acid. This messenger molecule then travels out of the nucleus and goes to the cellular factories, called ribosomes, where its instructions are read precisely as written, translating the code into the physical proteins that build the body. In this standard biological framework, the messenger ribonucleic acid is sacred. It is an exact, unedited photocopy of the original genetic code. If a species wants to change its physical proteins to adapt to a new environment, for example, growing thicker fur to survive an ice age, it must wait for a random, beneficial mutation to occur in the permanent deoxyribonucleic acid. This mutation is then passed down to the offspring, and over thousands of generations the species slowly evolves. This is Darwinian evolution. It is reliable, but it is incredibly, painstakingly slow. The octopus does not have thousands of generations to wait. It does not have time for the slow grind of standard evolution, so it simply breaks the central dogma. When the octopus creates a strand of messenger ribonucleic acid from its genetic code, it does not send that photocopy straight to the cellular factories. Instead, it intercepts the messenger. Using a highly specialized suite of biological enzymes, the octopus actively edits the temporary genetic code on the fly. This process is known as ribonucleic acid recoding. Specifically, the enzymes target a chemical base called adenosine within the messenger molecule and forcefully alter its atomic structure, converting it into a completely different chemical molecule called inosine. Because the cellular factories read inosine as if it were a completely different biological letter, the resulting protein that gets built is fundamentally different from what the original deoxyribonucleic acid actually commanded. To understand just how alien this is, we have to look at the scale. Humans and other mammals actually do possess the biological machinery for ribonucleic acid editing, but we use it incredibly sparingly, and usually only for highly specific immune system functions or minor neurological tweaking. In the entire human brain, we might edit a fraction of 1% of our messenger molecules. If a human cell edits its ribonucleic acid too aggressively, it usually results in catastrophic biological errors, leading to severe neurological diseases or cancer. But when molecular biologists analysed the nervous system of the octopus, they found a system running completely out of control in the best possible way. The octopus actively edits over 60% of the ribonucleic acid transcripts in its brain and nervous system. Scientists have mapped tens of thousands of specific recoding sites across the octopus nervous system. They are quite literally intercepting millions of microscopic messages every single second, crossing out the original genetic instructions and rewriting the code before it can be built. Why would an organism undertake such a massive, metabolically expensive, and biologically risky process? The answer is real-time, immediate environmental adaptation. Consider the vast dynamic environment of the ocean. An octopus might spend its morning hunting in the warm, sunlit shallows where the water temperature is highly elevated. But later in the afternoon, to escape a predator or chase a specific type of prey, it might plunge thousands of feet down into the deep, abyssal trenches where the water approaches freezing temperatures. For a highly advanced biological machine, temperature swings of this magnitude are usually fatal. In a complex nervous system, neurons communicate by firing electrical signals, and those signals are regulated by microscopic protein gates called potassium channels. In warm water, these protein gates open and close at a specific rapid speed, allowing the brain to function normally. But if you take those exact same proteins and plunge them into freezing water, thermodynamics takes over. The cold causes the protein structures to stiffen and lock up. The potassium channels close too slowly, the electrical signals misfire, and the nervous system completely crashes. This is why most highly complex marine animals must strictly regulate their internal body temperatures or stay within very specific thermal zones. The octopus, however, cannot regulate its internal temperature. It is entirely at the mercy of the surrounding water. If it dives into the freezing abyss, its brain should technically stop working. But it does not because of ribonucleic acid recoding. As the water temperature begins to drop, the octopus's cellular machinery instantly detects the thermal shift. Within hours, it massively ramps up its editing enzymes. It targets the specific messenger ribonucleic acid responsible for building those vital potassium channels in its brain. It edits the genetic code on the fly, physically changing the molecular structure of the resulting proteins. It custom builds a completely new variation of potassium channels that are structurally looser and perfectly optimized to open and close rapidly in freezing water. The octopus literally rewires the hardware of its own brain in real time to survive an environment that should have killed it. When it returns to the warm surface waters, it simply stops editing those specific messengers, and the brain smoothly transitions back to producing the warm water proteins. This is an unparalleled biological superpower. The octopus does not need to wait thousands of years for its deoxyribonucleic acid to mutate and evolve cold-resistant genes. It maintains a single, highly flexible base template of deoxyribonucleic acid and uses massive, widespread ribonucleic acid editing to dynamically customize its biology on a day-to-day or even hour-to-hour basis. It is effectively downloading software updates for its physical body to perfectly match whatever environment it finds itself in. But in the unforgiving economy of nature, every superpower comes with a brutal, unavoidable cost. By embracing this extreme form of real-time adaptability, the octopus has triggered a devastating evolutionary trade-off. It has essentially trapped itself in a biological dead end. To understand this trap, we have to look at the mechanics of how the editing enzymes know where to make their cuts. The enzymes cannot just randomly alter the messenger ribonucleic acid. They require a highly specific landing pad. For the enzyme to attach and change the adenosine to inosine, the strand of messenger ribonucleic acid must fold back on itself, creating a complex three-dimensional physical structure known as a double-stranded hairpin loop. This intricate physical fold is dictated entirely by the surrounding sequence of genetic letters. This means that if the octopus wants to keep the ability to edit a specific gene, it cannot just protect the single chemical letter being changed. It must aggressively protect the hundreds of surrounding letters that form the structural landing pad. If even a single, random mutation occurs in the underlying deoxyribonucleic acid of that surrounding sequence, the resulting messenger ribonucleic acid will not fold correctly. The structure will collapse, the enzyme will not be able to land, the editing process will fail, and the octopus will lose its ability to adapt. Because the octopus heavily edits tens of thousands of genes across its entire nervous system, it requires tens of thousands of these massive, complex structural folds. Therefore, the majority of its genetic code must be kept absolutely pristine. The octopus is subjected to an extreme form of what evolutionary biologists call purifying selection. The organism actively rejects almost all permanent mutations to its deoxyribonucleic acid, because any change threatens to destroy its precious ribonucleic acid editing system. The evolutionary implication of this is staggering. By relying so heavily on short-term ribonucleic acid recoding, the octopus has essentially frozen its long-term deoxyribonucleic acid evolution. While mammalian genomes are constantly mutating, shifting, and slowly inventing entirely new biological structures over millions of years, the genome of the octopus is essentially locked in place. From a strict genetic standpoint, their underlying deoxyribonucleic acid is evolving at a fraction of the speed of almost every other animal on the planet. They have sacrificed the long-term, slow-burn genius of Darwinian evolution for the immediate hyper-agile supremacy of real-time genetic editing. They are the ultimate biological paradox. They possess the most advanced, highly sophisticated invertebrate nervous system ever discovered, capable of tool use, facial recognition, and complex problem-solving. They operate biological cloaking devices that bend the physics of light, yet they achieve all of this using a genetic blueprint that actively refuses to evolve. They have hacked the most fundamental rule of molecular biology to conquer the oceans, painting themselves into a magnificent, highly intelligent evolutionary corner. It is this sheer, unprecedented defiance of terrestrial biological norms that leads us to the most controversial mind-bending question in modern science. If their genetic operating system is this alien, is it possible that they did not originate on this planet at all? When you combine all of these profound biological anomalies, the decentralized nervous system, the localized intelligence of the severed arms, the three-hearted circulatory engine-pumping copper-rich blue blood, the optical physics of a color-shifting skin driven by a colorblind brain, and a heavily scrambled genetic code that constantly rewrites its own messenger, ribonucleic acid, it becomes incredibly difficult to reconcile the octopus with the rest of the terrestrial animal kingdom. They are so fundamentally divergent from the standard biological operating procedures of planet Earth that they force us to look upward, past the surface of the ocean and into the cosmos. This brings us to one of the most controversial, intensely debated, and highly sensationalized moments in modern scientific literature. In the spring of 2018, a peer-reviewed scientific journal called Progress in Biophysics and Molecular Biology published a sprawling 33-page research paper authored by a coalition of 33 independent researchers and scientists from around the globe. The paper was titled Cause of Cambrian Explosion, Terrestrial or Cosmic, and inside its pages these scientists made a claim so radical that it shook the foundations of evolutionary biology. They formally proposed that the octopus did not evolve on Earth, but was brought here from deep space. To understand how 33 credentialed scientists could put their names on a paper claiming that octopuses are literal extraterrestrials, we have to understand the foundational theory they were building upon, the hypothesis of panspermia. Panspermia is an ancient philosophical and scientific concept, championed in the modern era by prominent astrophysicists like Chandra Wickramasinghe, who was also one of the lead authors of the 2018 paper. The core premise of panspermia is that the universe is not a sterile void, but a vast, active biological delivery system. It suggests that the fundamental building blocks of life, and perhaps even fully formed microscopic organisms, exist abundantly throughout the cosmos. According to this theory, these biological payloads are trapped deep inside the icy cores of comets, asteroids, and meteors. Protected from the devastating radiation of cosmic rays by thick shields of rock and frozen water, these biological seeds drift through the vacuum of space for millions of years. When one of these icy bolides is captured by the gravitational pull of a habitable planet like Earth and crashes into its oceans, the ice melts, the biological payload is released, and the alien life forms begin to thrive and colonize their new world. For decades, panspermia was viewed strictly as a fringe theory, primarily used to explain how the very first single-celled bacteria might have arrived on a young molten Earth. But the authors of the 2018 paper took this concept and massively expanded its scope. They looked at the Cambrian explosion, the sudden, violent biological diversification that occurred roughly 500 million years ago, where almost all modern animal phyla seemingly appeared out of nowhere in the geological record. The authors argued that the standard Darwinian model of slow, incremental mutation was mathematically far too slow to account for this sudden burst of complex life. Instead, they hypothesized that the Cambrian explosion was directly triggered by a massive sustained bombardment of extraterrestrial comets, carrying highly advanced spacefaring genetic material, specifically extraterrestrial retroviruses. But then, the paper focused its attention specifically on the cephalopods, and the octopus in particular. The scientists pointed to the exact same biological anomalies we have just explored. They highlighted the massive, complex brain, the camera-like eyes that developed completely independently from vertebrate eyes, and the highly chaotic, massively expanded genome. They argued that the evolutionary leap from a simple, slow-moving ancestral snail to a hyper-intelligent, shape-shifting octopus was too great, too rapid, and too complex to have occurred naturally in the oceans of Earth. Their ultimate conclusion was breathtaking in its audacity. They proposed that the highly complex genetic code for the octopus was not assembled by the slow trial and error of terrestrial evolution. Instead, they suggested that entire, fully functioning cryopreserved octopus embryos were frozen solid within a massive icy comet passing through an alien star system. This comet then traversed the interstellar void, entered our solar system, and crashed into the Earth's oceans hundreds of millions of years ago. The frozen alien embryos thawed out in the nutrient-rich, primordial waters, hatched, and unleashed the ancestors of the modern octopus into our ecosystem. In their exact words, the octopus was a pre-packaged extraterrestrial genetic software update that was literally dropped onto the planet from space. When this paper was published, it instantly made global headlines. The media seized on the idea that science had finally proven the existence of aliens, and that they were swimming in our aquariums. However, the reaction from the mainstream scientific community, specifically evolutionary biologists, geneticists, and paleontologists, was immediate, fierce, and overwhelmingly negative. The pushback against the octopus panspermia hypothesis was a masterclass in scientific rigor, tearing the theory apart piece by piece using the very same genetic and fossil evidence the paper attempted to exploit. The first and most devastating refutation of the alien embryo theory comes from the basic biochemistry of the octopus. If a complex organism evolved in a completely different star system, under a different sun, in a different chemical environment, its fundamental biological building blocks would be radically different from life on Earth. However, when we break down the octopus at the absolute lowest molecular level, we find undeniable proof of terrestrial kinship. The octopus uses the exact same deoxyribonucleic acid architecture as every other living thing on this planet. It relies on the exact same 4 chemical bases, adenine, cytosine, guanine, and thymine, to write its genetic code. More importantly, it uses the exact same 20 standard amino acids to construct its physical proteins. If the octopus were truly a cryopreserved alien from another world, the statistical probability of it independently evolving the exact same complex biochemical alphabet as a terrestrial oak tree, a human being, and a deep-sea bacterium is mathematically zero. The second major counterargument destroys the idea that the octopus simply appeared out of nowhere without evolutionary ancestors. While we established earlier that the octopus leaves a frustratingly poor fossil record, because it lacks a hard skeleton, the genetic record tells a very clear, unbroken story of its ancestry. When geneticists mapped the genome of the California two-spot octopus in 2015, they did not just find a chaotic mess of jumping genes and ribonucleic acid editing sites. They also found ancient, deeply conserved genetic markers known as Hox genes. Hox genes are the fundamental architectural blueprints of animal life on Earth. They dictate the basic body plan of an embryo, determining where the head goes, where the limbs grow, and how the digestive tract is formed. The octopus possesses a specific set of Hox genes that perfectly links it to the broader evolutionary tree of the phylum Mollusca. Through advanced comparative genomics, scientists can trace the genetic lineage of the octopus directly back to simpler, shelled mollusks. We can see the exact genetic pathways where the genes, responsible for building a hard calcium carbonate shell in a clam, were slowly mutated, repurposed, and eventually silenced in the ancestors of the octopus. We can trace the evolutionary history of their complex camera-like eyes, proving that they were built using pre-existing terrestrial genetic components that were highly modified over millions of years of intense predatory pressure. An incredible example of convergent evolution, not extraterrestrial intervention. Even the most alien aspect of the octopus, its ability to massively edit its own messenger ribonucleic acid to dynamically adapt to temperature changes, is not proof of space origin. Evolutionary biologists point out that while the octopus uses this ribonucleic acid editing system to a staggering, unprecedented degree, the fundamental biological machinery to perform those edits exists in many other terrestrial organisms, including humans and fruit flies. The octopus did not bring a magical alien technology to Earth. It simply took a pre-existing, highly obscure terrestrial biological tool and hyper-accelerated its use to compensate for the loss of its protective shell. Mainstream science views the 2018 panspermia paper not as a legitimate biological breakthrough, but as a fascinating piece of speculative fiction dressed in academic language. It is widely considered a failure of the peer review process, a case where a group of astrophysicists attempted to solve a complex biological mystery without fully understanding the nuances of modern genomics and evolutionary biology. The scientific consensus is absolute. The octopus is an earthling. It was born here, it evolved here, and its complex anatomy is the direct result of hundreds of millions of years of brutal, unforgiving survival in the oceans of our planet. However, despite the harsh debunking of the panspermia hypothesis, the fact that 33 scientists felt compelled to write such a paper is incredibly revealing. It serves as the ultimate testament to just how magnificent and terrifyingly strange the octopus truly is. The creature is so intelligent, so physically bizarre, and so genetically divergent that highly educated professional researchers looked at it and genuinely believed that the only logical explanation for its existence was that it fell from the stars. While the octopus did not come from outer space, its existence on Earth is a profound gift to the field of astrobiology. When astrobiologists attempt to theorize what intelligent life might look like on the icy moons of Jupiter or Saturn, such as Europa or Enceladus, they can no longer just look at humans or dolphins as the only models for higher cognition. The octopus proves that profound problem-solving intelligence does not require a rigid skeleton, it does not require a centralized brain locked inside a skull, and it does not even require an organism to see the world in color. It proves that nature can build a conscious thinking entity out of soft tissue fluid dynamics, and a distributed network of decentralized nodes. The octopus forces us to broaden our definition of life. It teaches us that if we ever do find actual extraterrestrial intelligence swimming in the dark, subsurface oceans of a distant world, it probably will not look like us. It will likely look, think, and operate very much like the phantom of our own oceans. While astrobiologists look to the stars for answers about the origin of the octopus, mechanical engineers are looking directly at the animal itself, treating it not as a biological curiosity, but as a technological blueprint sent from the future. For the past century, human engineering has been defined by rigid mechanics. We build our machines out of steel, titanium, and hard plastics. We construct them using fixed joints, linear actuators, and heavy gears. This paradigm of rigid robotics is excellent for precision manufacturing in highly controlled environments, like assembling automobiles on a sterile factory floor. However, when we place these rigid, heavy machines into chaotic, unpredictable, or highly delicate environments, they fail dramatically. A traditional steel robotic arm cannot safely interact with a fragile coral reef, nor can it safely navigate the highly complex, tightly packed organic tissues inside the human body. To conquer these chaotic environments, human engineering needed a massive paradigm shift. It needed a machine capable of infinite flexibility, localized intelligence, and gentle but terrifying strength. It needed to completely abandon the concept of the skeleton, and for that, the scientific community turned to the undisputed master of soft mechanics, the octopus. This realization gave birth to an entirely new, rapidly accelerating discipline of engineering known as soft robotics. Unlike traditional robotics, which relies heavily on the laws of rigid body dynamics, soft robotics is governed by the principles of continuum mechanics and fluid dynamics, directly mimicking the hydrostatic skeleton of the octopus. Engineers began to replace steel gears and electric motors with highly flexible elastomers, synthetic rubbers, and silicone polymers. Instead of using electrical currents to drive mechanical joints, they began pumping compressed air or water through microscopic internal channels, perfectly mimicking the way an octopus squeezes its massive muscular hydrostats. The goal was no longer to build a robot that could punch through a concrete wall, but rather to build a robot that could pour itself through a crack in that wall, exactly as the octopus does. The most immediate and profound application of this biomimicry is the development of the artificial octopus arm. A traditional robotic arm might have 6 or 7 degrees of freedom, severely limited by its artificial shoulder, elbow, and wrist joints. An octopus arm, as we've established, has infinite degrees of freedom. To replicate this, roboticists designed cylindrical silicon appendages embedded with complex three-dimensional networks of pneumatic chambers. By selectively pumping air into specific chambers along the side of the synthetic arm, the engineers caused that specific section of the silicone to rapidly expand, forcing the entire arm to bend in the opposite direction. By alternating the air pressure across the different internal chambers, the robotic arm can twist, coil, elongate, and snake its way around highly complex obstacles. The implications of this technology for the medical field are nothing short of revolutionary. In modern medicine, minimally invasive surgery is often performed using rigid endoscopes or stiff surgical tools that must navigate the delicate winding pathways of the human digestive tract or cardiovascular system. These rigid tools frequently cause microscopic tissue damage and are severely limited in their maneuverability. Enter the soft robotic surgical tentacle. Using the octopus as a structural template, biomedical engineers are actively developing microscopic, highly flexible surgical arms that can be introduced safely into the human body. Because these synthetic tentacles are entirely soft, they can gently conform to the natural curves of internal organs without causing lacerations or internal bleeding. Once they reach the target area, they can dynamically stiffen by violently increasing their internal fluid pressure, allowing the surgeon to perform highly precise tissue manipulation or tumour removal. What the octopus uses to violently rip open a clamshell on the dark ocean floor is now being reverse engineered to safely save human lives in the operating room. Beyond the microscopic realm of the human body, massive versions of these soft robotic arms are completely changing how we interact with the deep ocean. Marine biologists have historically struggled to collect living samples from delicate deep-sea ecosystems. Traditional metal claws attached to remote-operated submarine vehicles easily crush fragile specimens like sea jellies, rare sea sponges, or ancient corals. Today, deep-sea exploration vehicles are being outfitted with large, highly sensitive silicon octopus arms. These soft manipulators can gently wrap around a fragile piece of deep-sea coral, evenly distributing the mechanical pressure across the entire surface of the object, capturing it perfectly without leaving a single scratch on its surface. But an octopus arm is only half of the mechanical equation. The true holding power of the animal comes from its thousands of independent suction cups. Designing an artificial suction cup sounds relatively simple, until you consider the extreme physics of the ocean floor. Human-engineered suction cups work perfectly on clean, dry, perfectly smooth surfaces like household glass or polished metal. But if you try to stick a standard plastic suction cup to a wet, slimy, heavily textured rock deep underwater, it instantly fails. The water and the microscopic unevenness of the rock completely break the vacuum seal. The octopus, however, can achieve a flawless, unbreakable grip on a slimy, jagged rock while simultaneously fighting against violent ocean currents. To crack this biomechanical mystery, materials scientists studied the microscopic architecture of the octopus sucker. They discovered that the edge of the animal's suction cup is not a simple, smooth ring. It is densely packed with highly flexible microscopic ridges and valleys that can physically deform to match the exact microscopic topography of the target surface, actively pushing away water molecules and creating a flawless, highly localized vacuum seal. Inspired by this, engineers have created octopus-inspired smart adhesives. By casting synthetic elastomers in microscopic molds, they have created materials lined with thousands of artificial microsuckers. Furthermore, they combine this structural design with advanced temperature-sensitive polymers. When these synthetic suckers are heated, they physically expand and lock onto a rough, wet surface with incredible holding force. When the temperature is slightly lowered, they instantly release their grip. This biomimetic breakthrough is currently paving the way for underwater construction robots that can walk vertically up the side of a submerged steel oil rig, and for advanced medical patches that can firmly stick to wet internal human organs during surgery without ever requiring chemical glues or damaging metallic sutures. Yet the absolute ultimate frontier of octopus biomimicry lies not in physical mechanics, but in optical physics. The scientific race to perfectly replicate the dynamic, shape-shifting and colour-changing skin of the octopus is one of the most heavily funded pursuits in modern materials science and military engineering. The overarching goal is to create synthetic active camouflage, a material that can instantly read its surrounding environment and fundamentally bend the physics of light to make whatever it is covering completely disappear from the visual spectrum. To achieve this incredible feat, engineers have developed thin sheets of hyperelastic dielectric elastomers, a specific type of smart synthetic rubber that physically changes its geometric shape when exposed to an electrical current. Embedded deeply within these rubber sheets are millions of microscopic, highly engineered pockets filled with black carbon-based liquid dye, acting as artificial chromatophores. In their resting state, these pockets are extremely tiny and completely invisible to the naked eye. But when a computer system fires a targeted electrical voltage across the sheet, the artificial rubber rapidly expands, stretching the pockets of carbon dye into wide, highly visible discs, instantly turning the transparent sheet pitch black. By layering multiple sheets containing different coloured dyes on top of one another, engineers have successfully created the world's first artificial, highly flexible, colour-shifting skin. but the illusion of invisibility is instantly broken if the surface is completely flat while the surrounding natural environment is highly textured. To conquer this optical hurdle, researchers looked directly to the octopus's papillae, the massive muscular bundles that allow the animal to grow complex spikes and bumps out of its smooth skin in mere milliseconds. In the laboratory, scientists have engineered advanced fiber optic meshes entirely coated in a highly specialized silicone matrix. By violently forcing compressed air through highly specific algorithmic patterns within the internal mesh, the two-dimensional silicon sheet violently erupts into complex three-dimensional shapes, perfectly mimicking the rough texture of rocks, the jagged edges of leaves, or the coarse grain of ocean sand. When you seamlessly combine the artificial color-shifting chromatophores with the three-dimensional synthetic papillae, the resulting material defies modern comprehension. Military defense contractors are actively developing these octopus-inspired metamaterials to coat military transport vehicles, stealth submarines, and even individual infantry armor. The concept is no longer science fiction. In the very near future, a military drone could land in a dense forest, instantly read the optical and physical texture of the surrounding trees using advanced camera sensors, dynamically shift its synthetic skin to perfectly match the brown and green hues of the bark, and physically grow artificial leaves, effectively erasing itself entirely from the battlefield. The octopus is effectively forcing humanity to completely rewrite the fundamental rules of mechanical and optical engineering. For thousands of years, we built machines based strictly on the rigid biological structures we saw in ourselves and the terrestrial animals around us. We built heavy skeletons of iron and artificial muscles of steel. But by closely observing this completely divergent, shell-less biological phantom of the deep, we have realized that the future of advanced technology is not hard, rigid, and binary. The true future of technology is soft, highly fluid, aggressively adaptive, and infinitely complex. The octopus represents the absolute pinnacle of what biological engineering can achieve over millions of years of brutal survival, and it serves as the ultimate, undeniable proof that the greatest technological blueprints in the entire universe are currently swimming completely unnoticed in the absolute darkness at the bottom of our oceans. We have painted a portrait of an organism that borders on the supernatural. We have explored a creature equipped with a massive decentralized neural network, a shape-shifting optical cloaking device, an engine of 3 hearts pumping heavy metal blood, and a completely chaotic, rule-breaking genetic code that rewrites its own biology on the fly. By all conventional logic of evolutionary biology, a creature possessing this terrifying arsenal of physical and cognitive adaptations should be the undisputed immortal apex predator of the oceans. You would expect an intelligence of this magnitude to live for decades, accumulating vast libraries of environmental wisdom, much like a 50-year-old elephant or a centenarian blue whale. But here, we crash into the most tragic, baffling, and cruel paradox of the octopus. For all of its alien brilliance, it is a fleeting phantom. The vast majority of octopus species live for only 1 to 2 years. Even the absolute largest of their kind, the massive giant Pacific octopus, reaching weights of over 100 pounds and arm spans of 20 feet, rarely survives past its 4th birthday. Why would nature invest such a staggering amount of metabolic energy into building one of the most advanced biological machines on Earth, only to throw it away almost immediately? The answer lies in a deeply unsettling biological phenomenon known as programmed senescence. For the octopus, death is not merely a consequence of slowly wearing out over time. Death is a strictly scheduled, biologically triggered, and chemically enforced self-destruct sequence, and it is directly tied to the single, most important event in their short lives: reproduction. For an octopus, to mate is to sign its own death warrant. The biological clock of an octopus is heavily monitored and controlled by a tiny paired organ, located directly between its central donut-shaped brain and its massive eyes, known as the optic gland. This gland acts as the master control switch for the animal's entire endocrine system. During the first phase of the animal's life, the optic gland produces the precise mixture of hormones required to keep the octopus growing, hunting, and learning at a rapid pace. But the moment the animal reaches sexual maturity and successfully mates, the optic gland undergoes a catastrophic, irreversible shift in its chemical production. For the male octopus, decline begins shortly after he transfers his genetic material to the female. His optic gland alters its hormonal output, and he enters a state of rapid physical and cognitive decline. He stops hunting. He loses his ability to flawlessly coordinate his chromatophores, causing his brilliant camouflage to fail, leaving him wandering aimlessly and highly visible in the open ocean. Within a matter of weeks, he either starves to death or is easily picked off by a predator. But for the female octopus, the self-destruct sequence is far more dramatic, terrifying, and purposeful. After mating, she actively seeks out a dark, hidden den deep within a rocky crevice. There, she lays tens of thousands of tiny teardrop-shaped eggs, meticulously weaving them into massive cascading braids that she glues to the ceiling of her den. From the moment the last egg is laid, she will never leave that cave again. The optic gland violently shifts into overdrive. Recent biochemical studies into this exact moment have revealed the horrifying mechanics of this process. The optic gland fundamentally rewrites the animal's cholesterol metabolism. Instead of using cholesterol to produce standard healthy hormones, the gland begins to synthesize massive quantities of specific steroid hormones and a highly toxic chemical precursor called 7-dehydrocholesterol. This sudden flood of modified chemicals acts as biological poison. It forcibly shuts down her digestive system so she completely loses the desire to eat. It then triggers a rapid, systemic degradation of her own cellular tissues. As she spends the next several months gently blowing fresh, oxygen-rich currents of water over her eggs to keep them clean and healthy, her body is literally digesting itself from the inside out. She uses her own dissolving muscles to fuel the energy required to protect her brood. The physical transformation is agonizing to witness. Her vibrant, colour-shifting skin turns a pale, sickly white and loses its complex texture. Her massive, powerful, muscular hydrostats wither away into thin, weak ribbons. Her eyes cloud over. In the final, most extreme stages of this chemically induced dementia, the toxic hormonal flood is so severe that females have been observed actively tearing off their own skin or biting the tips off their own arms. She defends her eggs with relentless, starving fury until the exact moment they hatch. As tens of thousands of microscopic, perfectly formed baby octopuses float upward into the ocean currents, the mother finally succumbs to her programmed degradation and dies on the floor of the den. From a human perspective, this seems like a colossal evolutionary mistake. Why build a genius brain just to poison it? But evolutionary biology is completely devoid of sentimentality. It only cares about the survival of the species. Biologists theorize that this brutal self-destruct mechanism is an ecological necessity. Octopuses are voracious, highly aggressive, and completely solitary apex predators. If the older, massive, highly experienced adults were allowed to live indefinitely alongside the newly hatched young, the ocean simply could not support their collective appetite. More importantly, because octopuses are highly prone to cannibalism, the immortal adults would likely consume the entire next generation of their own species. By chemically forcing the adults to die immediately after reproducing, nature guarantees a massive, constant turnover of generations. This rapid turnover is exactly what allows their chaotic genome, driven by jumping genes and ribonucleic acid editing, to constantly shuffle and adapt to the changing oceans, without the older generation monopolizing all the resources. But understanding the mechanics of their death inevitably leads us to the realm of speculative biology, and forces us to ask the most profound what if in the animal kingdom. What exactly separates humans from the rest of the natural world? It is not merely our intelligence. It is our capacity for intergenerational knowledge transfer. Because humans live for decades, we have the time to raise our young, communicate our experiences, and teach them everything we have learned. A human child does not need to figure out how to build a fire from scratch. They are simply taught how to do it by their parents. We build culture, technology, and civilization by stacking the knowledge of one generation on top of the next. The octopus is entirely denied this luxury. Because the parents die before or exactly when the offspring are born, every single baby octopus is an orphan. They inherit a massive, highly capable brain, but zero accumulated knowledge. Every single time an octopus figures out how to unscrew a glass jar, how to build a shield out of coconut shells, or how to short-circuit a laboratory light bulb, it has figured it out entirely on its own through isolated trial and error. All of that brilliant, hard-won experience dies with them after 1 year. They are born into a biological vacuum, forced to invent the wheel from scratch, generation after generation, millions of times over. Now, imagine a theoretical timeline where a simple genetic mutation suddenly disables the toxic cholesterol pathways of the optic gland. Imagine if the biological self-destruct sequence was turned off. What would happen if an octopus lived not for 2 years, but for 50 years? If an animal with 9 brains, a decentralized nervous system, and the ability to manipulate objects with infinite mechanical freedom were given the time to accumulate 5 decades of memories, the result would be terrifying. If they lived long enough to overlap with their own offspring, they might begin to recognize the survival value of teaching. An older octopus could demonstrate the precise technique for dismantling a crab trap or navigating a specific reef to its young. This would require communication, and with their highly advanced color-shifting, shape-shifting skin, they already possess the most sophisticated visual communication array on the planet. They could literally flash complex, moving images across their bodies to convey detailed information to one another. With the barrier of the short lifespan removed, intergenerational knowledge would begin to accumulate. Solitary behavior might slowly give way to coordinated social hunting to take down larger prey. They could move from merely hiding in discarded coconut shells to actively farming and building permanent, complex underwater structures using rocks, shells, and kelp. Without the biological hard limit of their optic gland, the octopus would cross the threshold from a solitary intelligent animal into a culturally and technologically advancing species. They would possess all the necessary physical and cognitive hardware to establish the first non-mammalian civilization on Earth. The tragic reality is that the only thing keeping the octopus from rivaling humanity for dominance of this planet is a microscopic chemical switch located between its eyes. They are a species held back only by the relentless ticking of a biological clock. They are brilliant alien phantoms trapped in a fleeting existence, masters of an underwater universe they will never live long enough to truly conquer. The octopus may not have fallen from the stars inside a frozen comet. Its genetic roots are irrevocably tied to the evolutionary history of planet Earth. Yet, it stands completely alone as the ultimate biological outlier, a magnificent anomaly that defies almost every single rule we have established for complex life. By abandoning the safety of a hard, calcium-based shell, it forced itself to become a creature of pure liquid intellect. It built a decentralized mind, distributing its consciousness across 8 tasting, feeling, and thinking arms. It engineered a circulatory system fueled by copper and powered by 3 distinct hearts. It mastered the physics of light, turning its very skin into a complex shape-shifting display that can render it completely invisible in a fraction of a second. And it learned to hack its own genetic code, actively rewriting its messenger ribonucleic acid in real time to conquer the most extreme environments our oceans have to offer. They are not extraterrestrials, but they are the absolute proof that the evolution of intelligence did not follow a single narrow path. Humanity represents the pinnacle of the vertebrate model, a centralized brain locked safely within a rigid skeleton. But the octopus proves that there is an entirely different biological operating system for consciousness. It shows us that profound, problem-solving awareness can arise from soft tissue, decentralized neural networks, and beautifully chaotic genetics. They are a fleeting glimpse into an alternate reality, a brilliantly intelligent species tragically constrained by a brutal, chemically programmed expiration date. They remain the beautiful, tragic phantoms of the deep, experiencing the universe in a way we will never truly comprehend. If you found this deep dive into the alien biology of the octopus fascinating, please consider leaving a like and subscribing to the channel for more scientific explorations. And before you go, I would love to hear from you. Please leave a comment below letting me know where in the world you're watching from, what time it is, or what you think our planet would look like if the octopus lived for 50 years. Thank you for watching.